Dushan, how are you?
Good. How are you?
Sorry, my mic was not working before.
It took a while to.
Yeah.
I see Dr. Chandani is also there.
Oh, hi, Dr. Chandana.
Okay, I guess we'll start and people may join as we continue. So good evening, everyone, and welcome you all to the 4th research seminar happening at the Department of Computer Engineering, University of Peradeniya. I welcome you all to our seminar today. And as per the speaker, we have Dr. Dushan Wadduwage. And let me take a few minutes to introduce Dr. Dushan Wadduwage to everyone. So Dr. Dushan Wadduwage is currently a John Harvard Distinguished Science Fellow in Imaging at Harvard University. And he also leads the Wadduwage Lab. So I'm going to quote from their description. So Wadduwage Lab is working on novel computational microscopy solutions that can measure biological systems at their most information-rich form with a minimum redundancy. And their approach is said to enable faster measurements, higher throughput, and sometimes novel experiments. So that I directly quoted from their description. And to briefly present his journey, Dr. Dushan Wadduwage holds a PhD in biological systems from the National University of Singapore, and he holds a Bachelor's of Science in Electronic and Telecommunications Engineering from the University of Moratuwa. And prior to joining the Harvard University, he has been a postdoctoral associate at MIT, USA, and also, So, Dr. Dushan, we are delighted to have you today as a speaker. We saw your recent work that was featured in the news as well. So we look forward to hear about your work and the work that is done in your lab. So over to you.
Thank you. Thank you so much, Damayanti. And first of all, thank you very much for inviting me to give this talk. I'm really excited to be here. And also thank you everyone for attending this. I hope it's not going to be that boring, although I'm sure it will be a little boring to some of you in some of the places. So let me share my screen. Start with. Can you all see my screen?
Yes, looks good.
Okay. Very good. Yeah, so today I'm going to talk about like a family of like approaches that we have been developing in my lab. We call differentiable microscopy. And we do this for like to get high throughput imaging or like high throughput microscopy applications. So like the quoted like, so the idea behind my lab is that we try to design optical instruments like microscopes, as well as the algorithms to process data acquired from these instruments together. So we do both of these aspects in-house. And this allows us to like incorporate some of the information about the algorithm in the design stage of the instrument itself, and also some of the information about the instrument in the design of the algorithm. So like treating this thing as like a holistic process, like we believe like we can do better instrumentation. So that's sort of like the idea. And I have this space like turn it up to 11. If you have time at the end, and if you guys like reach the end, wait until the end, like I'll probably show you like why it's there, where the reference comes from. Yeah. So first, let me like give you an idea of like why we do this kind of instrumentation and why we care about like imaging speed. So can you also see my mouse pointer?
Yes.
Okay, sounds good. So basically, like here, what you see is a fluorescence image of a cell, the cell nucleus. So in blue, what you see is the nucleus, and it's about like 10 micrometer in size. And then what you see in this green, on these green dots, are actually DNA damage and repair events. So what happens is like when our cells are exposed to DNA damaging agents, they're going to, our DNA is going to be damaged, and then there is like internal mechanisms in the cell that can actually correct these damages. So if the DNA is damaged, like it can lead to cancer, right? So it doesn't happen because like we have these mechanisms to correct this. So this is an experiment to measure how these mechanisms kicks in, like how much DNA damage is there, like what happens. So basically what we are interested in this sense is looking at these individual spots and like maybe the number of spots per cell and like how they are distributed, how they change over time, like things like that. But to do that, we need to be able to image like at really high resolution because we need to see these individual spots and we need to be able to resolve them. On the other end, we also have to image like hundreds of thousands or like even millions of cells in order to build the statistics for this kind of experiment. So now you can see like we have these two like competing interests. We need to be able to do high resolution images, and also we need to be able to image like a really large area or a volume. So this case tries to a problem of like measurements. So just to give an idea of like the problem quantitatively, let's say like I want to image this 1 centimeter by 1 centimeter by 1 millimeter volume. And I want to see like here we saw like there are two types of information in the blue and the green channel. And like, let's say I want to do like 4 channels, like 4 different types of things in the cell. And I want to image this at this like resolution 0.25 micron by 0.25 micron by maybe like 1 micron. So this is the voxel size. So if to take this measurement, we're going to have to take around 16 trillion, like we are going to have to measure about 16 trillion voxels, which is a lot of like measurements. Now, in fact, if you use like a normal point detector, which measures like 1 point at a time, to complete this measurement, even with like the fastest detectors we have today, it's going to take around 5 years. So this is like clearly not feasible, right? But luckily, we have access to these systems that we call wide field detectors. These are the detectors that we have in our cameras and like most other places, which has like millions of pixels or sensors on a single chip. But they can only be operated at lower speeds. But even at that speed, like you can just, due to the parallelism, like you can complete this measurement in like 11 hours. Okay, so 11 hours is feasible, 5 years is not. But what happens at the data side? So if I complete this measurement, then this is going to give rise to about 32 terabytes of data, which is reasonable, but like you want to be able to do these experiments like regularly. So this is quite a bit of data, and like let's say I'm really cheap to buy that much storage. So what I'm going to do is like I'm just going to compress that. So let's say I compress it into 100 times. Now, if I compress it into 100 times, then I end up with 320 gigabytes of data. And I spent like 11 hours. So 11 hours, 320 gigabytes, this is not bad. So this is definitely like feasible and doable. But if you look at this process like a little carefully, you can see that there is an issue here. I spent 11 hours acquiring 32 terabytes of data, but just to end up having only 320 gigabytes of useful information. So like I spent way more time like measuring redundancies. What if I can just measure this 320 gigabytes of information itself without having to measure this massive like 32 terabyte data set? then I would actually be able to complete this measurement in like 6 minutes, which is way more attractive. So that's kind of like where we are in the systems that I'm going to talk about today, and also like most of the systems that we develop in the lab. We are trying to measure these imaging signals in some sort of their compressed form, so that like we have an advantage in like acquisition time. Yeah, so with that, I'm going to talk about like 3 things in today's presentation. So first, I'm going to kind of like give you an introduction to computational imaging, or like computational microscopy. And then then I will talk about the recent paper DEEP, which stands for descattering with excitation patterning, and that is built on top of like using these computational imaging principles. And that would kind of like work as an example to show like how this theory come into practice, and then what you can do about doing this. And then finally, I will talk about the approach that we use to like optimize these instruments, or like make these instruments smarter, called differentiable microscopy. So that's where like we kind of like try to learn some prior information from using deep learning and like machine learning approaches to optimize the instrument further. Yeah, so with that, like let's go to the first part, the computational imaging, or like computational microscopy. Now here I'm trying to like introduce the problem in a like some abstract sense. So let's say I want to take an image X, and now the image X is like represented as a vector n into 1. So the number of pixels in X is like the image is capital N, and it's a 2-dimensional signal, of course, but like you can just column stack it up and then like make it linearized, and then you will end up with like a vector. And then you have this optical system A, which we also call the forward model, to kind of like translate that signal, or like project that signal into the measurement space. Now the measurement space is represented by Y. And the idea is like Y is also a vector, but that is m to 1. And m is smaller than n. So like we take like lower number of measurements than the number of pixels here. So once you do that, you need to kind of like reconstruct the image again using Y. And that has to be done through some sort of like an algorithm, or like solving some equations, which is like broadly represented by A inverse here. And then you can get an approximation for your original signal X, like which is represented by X hat. Now, everything in optics, like at least the ones that we're talking about today, is going to be like linear. So if it's linear, like you can you can see that this can be represented as a set of linear equations. So the unknowns are the pixels, or like the entries of X, and then the measurements are the entries of Y. And they're going to be related to each other using a measurement matrix A. So this is like a linear system in the form of a matrix equation. So if I expand it further, like let's say like now we're trying to do like m is less than n. So like this is going to be a smaller vector than the original measurement vector X. And then that's A is some sort of a matrix that kind of like relates these two. The problem is like if I try to do it, if I want to solve this kind of system, like the most straightforward way of doing that is like just solving the inverse, right? So I have the X hat equals A inverse Y, but technically you can't do this here, because this is an underdetermined system, because you have more unknowns than the number of equations. So you need to play some tricks in order to solve this system. So that's where this one of the core concepts of computational imaging comes into play, which is called compressive sensing. So basically what compressive sensing says is the following. I have my X, but this is a naturally occurring signal. It's not an engineered signal. So if it's a naturally occurring signal, the hypothesis is that every naturally occurring signal has redundancies. So the only thing that doesn't have redundancies is like pure noise, which we are not interested in. So if it has redundancies, then it can be represented in some compressible form. So here is like the compressible form is like psi alpha. Now psi is some sort of a matrix that projects X back into this alpha, or vice versa. And the idea is like if X is compressible, if we can find this correct psi, then alpha has like most of the entries of alpha is like either very small or 0. And there are only like large non-zero entries of alpha that corresponds into the signal. And now if I can write this, I can write like if this is true, then I can just convert my linear system like this y equals a into psi alpha. Now to just show that like this is true, so the images are usually like we call sparse or like compressible in the wavelet basis. So this is some wavelet basis, and then like if I take all the wavelet coefficients to reconstruct the image, this is how it's going to look like. But if I only take like the 1% of the most like non-zero entries, then this is how the image is going to look like. And like the 99% of the other ones I can just throw out and still get a reasonably like accurate image. So this is like an like example showing that like the image signals are compressible in the wavelet basis. So like this psi is some sort of like a wavelet matrix here. Now, if you can do that, if you find this basis that is sparse in, the compressive sensing says you have multiple solutions because like you are an underdetermined system. So basically, like if I do it in some hyper like a high dimensional space like this, there is a plane or a surface that's kind of like gives you all the solutions. Like here, it's like just drawn in 2-dimensional space, and then this entire line is actually have the solutions that satisfy this equation. But like what compressive sensing says is like, okay, so this has to be sparse. So if you find the sparsest solution, that's going to be your right solution with overwhelming probability. And to find that, you can actually do this this optimization just by like doing minimizing the L1 norm of alpha with constraint to this equation. That looks like this diamond here, and then if you start minimizing it, and then the point that it kind of like agrees with this like is going to be your solution. So here you can see like alpha 2 is non-zero, but alpha 1 is 0. So it's a sparse solution. So this seemed to be working like reasonably well. In practice, we have some noise and like more complications, so you don't really do that optimization. What we do is like an optimization like this nature. So we have this, this is the L2 norm of the equation, and we try to minimize the combination of L2 norm with L1 norm of the alpha with some kind of like a regularization weight. So this usually works well. And here what you see is like using this this method in a real experiment called single pixel camera. It's a computational imaging system, like one of the first that was built. So this is the uncompressed version of like this letter R, and then if you measure it like 50 times compressed version of that, like you can still see an R. So basically like this is by solving this underdetermined system using this kind of like an optimization algorithm. So the same idea has also been translated to microscopy. And this is one of the first systems that was like demonstrated using this kind of approach. And what you see is here is like 8 times compressed image of this like uncompressed image. So, so far I talked about like the mathematical framework behind compressive sensing or like computational imaging. But how do we actually go about building these systems? So I'm going to take this as an example. This setup, which is called a single pixel camera. And this is one of the first demonstration of this idea. So what we have here is a camera, but it has only one sensor or like a single pixel. So how it's going to measure an image is by trying to take like multiple measurements using this sensor over time, and then it's going to reconstruct the image using these multiple measurements. So in the setup, you have a light source here, and then this is the object, and this object is going to be illuminated with this light source. And then there's like a lens and lens 1, which is going to image the light source into some image plane. So usually in the image plane, we place a camera, but now here what they have done is like they placed this device called the DMD. So DMD is called a micromirror device. That's like the active element in the projectors. And it has like millions of very small mirrors. And if you turn these mirrors in, so they can be turned on and off. So if you turn them on, like the light is going to be like go in this optical axis. And if you turn it off, like the light is going to go somewhere else. So basically, it can be used to turn on and off pixels of the image of this object. Now, let's say like we switch on pixels on the DMD according to some pattern. And then that is all the light that comes from that image, now modulated with that pattern, is going to be like collected using this lens 2, and then going to be like measured using this photodiode circuit, or this is the single sensor. Now, if you look at this in terms of this equation, like now X is the object here, and the first row of A is actually contains the information about this pattern on the DMD, like how the pixels are switched on and off. And then it's going to have like this dot product, right, in this matrix equation. And then that dot product is a scalar. It's going to be the first element of Y. Now, how we build up the measurement is like you're going to project a second pattern, and then that is going to be the second row of A. And then you're going to take the second measurement, that is the second entry of Y. So likewise, you can keep building up this linear system. And when it is underdetermined, you can still solve this using the optimization that we talked about earlier. So I believe that this gives you kind of like a clear idea about how these systems work. So before going into the next phase, like if you have any questions, I'd be happy to like answer. This can be a discussion rather than just me talking. So yeah. So if any of you have questions, like please feel free to ask. Okay, sounds good. So next, I'm going to kind of like explain how we use this idea to build a microscope in the lab. So we build this system to do like what we call the descattering with excitation patterning, and it operates in an imaging modality called two-photon fluorescence microscopy. So before I explain that, like I'll first give an introduction into like how fluorescence imaging work. Now the target of our experiment is to, let's say, like measure like how the brain operates in like a mouse, let's say. So my collaborator Elena Devi from MIT Neuroscience, so she has these experiments, like so her lab actually make this genetically modified mouse, which express fluorescent proteins, like fluorescent molecules, in some parts of the brain. Now, what we want to do is like we want to see like how these where these molecules are. So the position of these molecules are the ones that we want to measure. So since they are fluorescents, what happens is like if I, for an example, send blue light, these molecules are going to go into, so this is the ground state, and then it's going to absorb a photon of like blue light, and it's going to go into a higher energy state here. Now in the higher energy state, because it's unstable, like it's going to lose some energy, and then later it's going to kind of like come back to the ground energy state. When this happens, it's going to emit a photon in a different color. Like if you excite with blue, then it's going to emit, like for example, light in green. So as we know, like blue has a higher energy, blue photons have higher energy. And since you lose some energy here, like the photon that's emitted is going to be lower energy, and then that's going to have like a longer wavelength. That's why it's in green. Now this is interesting because now I can, if I have a way of like filtering out the blue photons, like basically if I have a filter that rejects all the blue light, and if I just pass through the green light, then I can see like where the molecule is, right? That's what we are interested in. So this is like a very powerful way of doing imaging, because you can actually make specific molecules express proteins, and we can measure their location using imaging using this technique. Now, what will happen if I send red photons? Let's say like I use two red photons, which in combination have the same energy as the blue photon. Now let me send it. Okay, nothing happened. Try it again. Nothing happens. Try it again. Okay, so what happens is like once in a while, if these photons satisfy some specific properties, this fluorescent molecule can actually absorb them and go into the same high energy state. So absorbs, like instead of absorbing one photon, it's going to absorb like two lower energy photons and go into a high energy state. And then the next process is like almost exactly the same. It's going to lose some energy and then come back down to the ground state, emitting green photons. Now, why do I want to like I showed in the demonstration or like the cartoon, this is like way less efficient than just doing like a blue photon. So why do I want to do like these red photons? The reason is the following. So this is actually an image of my hand. So I took this image yesterday. So this is like a light illuminator or a light from an iPad, and then like I'm just putting my hand there, and this is like bright white light. But through the hand, I can only see like the red light comes out, right? So this is because of a specific property of the light. When the wavelength of the light is lower, it's going to see like less scattering. So in other words, if I have like these red photons, they are going to see like less scattering. So they are going to be I will be able to transmit them through scattering medias like the brain, like tissue. So I can actually, the motivation of using this two photon excitation or like the red light to the excitation is because I can, like, without scattering, I can penetrate through tissue, and I can image much below the surface and see like what's happening below the surface of the brain. So like these experiments are done on like live animals. So this is like really interesting. Yeah, so with that, so this is what we want to do. We want to like image through scattering media using two photon excitation. And this is like the status quo. Many labs in the world, like right now, using this technique called point scanning to photon microscopy. And what they do is like they just image one point at a time. And you can actually like reconstruct this like nice fitting images. This is an image of a pyramidal neuron, and what you see here in like bright like large spot is called soma, or the cell body. And then this is like a dendritic branch. And what we are interested in is small protrusions, which are called the spines. This is actually very important to see like how neurons kind of like connected to each other. So only through these like locations, one neuron is connected to each other, and then like therefore the connections form and this associate. So it's interesting to look at these positions and how they develop throughout the lifespan of the animal. So that's using point scanning. And if I can do the same experiment in wide field and use a camera to do the detection, and like we said, like it should be in theory like much faster, because like you have multiple sensors, like millions of sensors that can be used. But the image quality is going to be like very low compared to point scan. So the reason is the following. Now, what happens here is we can send red light, which excite molecules through scattering mediums without anything happening to them. And if, in fact, like if I send these letters like MIT through scattering medium into like a fluorescence kind of like molecules below, and if I image from the bottom, then I'm going to see these like nice letters. And we have in fact done this experiment, and you can see the nice letters. But if I image it again in the reflective mode in the other side, I'm going to see this like very blurred image. So what happens is I can actually send the red photons forming this pattern of like letters. But when they come back to the scattering tissue, the green photons are coming back, right? So because the green photons have much shorter wavelength compared to red photons, they are going to scatter more. So like I see a blurred image. So to address this problem, so what we thought of, like, we want to be able to use the wide field sensors to do this measurement, because like then you can operate much faster than the point scanning. So to address this, what we thought is like, okay, so like similar to single pixel camera, we will project some sort of a pattern in this. And we know that the pattern is gonna go as we send it, because it doesn't see much scattering because it's in red light. And the image that we are gonna measure again is gonna be scattered, but now it's going to have some modulation based on this excitation pattern. So like intuitively, you can see like we thought of like, okay, so you send many of these patterns and then you collect many of these images, and I can use the information about the patterns and then the images to kind of like reconstruct a clear looking image. And it's still going to be faster because the number of patterns I have to use is much less than the number of pixels that you normally scan using point scanning measurement. So before I show like how it works, I'm going to kind of like briefly explain the optical setup in the most higher level possible. So what we have is like we have a specialized laser which emits like pulsed light at very high energy, or like very high energy per pulse, and then this light is gonna like go through what we call a grating. So what happens in the grating is like one pulse comes through, but because of the grating, it's gonna like spread out. And then we are going to image that pulse back into what we call a DMD. Like that's the same device which we saw on the single pixel camera. Now the DMD can actually project some sort of a pattern into the focal plane of the sample. There's a reason why we use this grating. It's called the temporal focusing, but I'll not talk about this, and it's not like relevant for the things that we discussed, that I want to discuss today. But there's a reason why we do that. So basically what happens is like on the focal plane, I can actually use this DMD to switch on and off pixels as I see fit. And then the emission light scattered is going to come through. And this mirror actually kind of like reflects the red lights and then let the green light pass through. And then I'm going to collect that light and then image it into a camera. Now this is not specifically a single pixel microscope or like single pixel camera, because I have multiple cameras. But the idea is like every pixel in the camera can be kind of like thought of as a single pixel detector itself. So like you have the parallelized, much scaled up version of a single pixel imager. So that's like the idea, and you can form that mathematically in this equation. So what happens is like X is our image. Now it's like represented as XY, like two-dimensional signal. And then this HT is going to be the teeth pattern. And this XPSF is called the excitation point spread function. It's also a function of XY. This, you can think about this as the impulse response in the excitation side of the system. And it's going to convolve with the pattern, and then this is the this whole thing is the effective pattern that we will project into the sample. This is like HT is the pattern on the DMD, and then this whole thing is the pattern that's going to be projected onto the sample, and then it's going to multiply with the object XY. And then the whole thing is again going to be convolved with another point spread function or an impulse response at the detection site. So this is we call the scattering point spread function, and it contains all the information about the scattering and how the light travels through scattering medium, and also like information about the collection optics. Now, if you look at this, like it can be some, it's much more complex than that, but like it is something like a Gaussian. So if this is something like a Gaussian, and first of all, like let me show you like what will happen if you don't have all these pattern excitations and everything. So let's say like I have an object 1 and 2, like D1 and D1 here. D1 has lower frequency. So like this is low frequency means like low resolution information, and E1 has like only high resolution information. Now, if I do imaging using my setup without any patterning or anything, then I'm going to see an image corresponding to D1 here. But the E1, I'm not going to see an image at all. This is because like this scattering point function actually acts as a low pass filter. So only the low frequencies are passed through, and then high frequencies are just going to completely filtered out. And if you think about it in the Fourier domain. What happens if I include the patterns? Now the EXPSF is like almost an impulse. And then we have this pattern, like let me take some sort of like a random pattern. Now if I image, I'm going to see like really a messed up image here. But the important thing is that I'm going to see images for both of them. So basically this shows that like we can actually pass this high frequency information by using the patterns. So in other words, like more technical way of saying it is like the high frequency information in the signal X is going to be aliased back to the passband of the scattering point function. So even this act as a low pass filter, some of the high frequencies are still going to pass through that. So it's going to be messed up image, but like I can measure the high frequencies. So with that, like we can actually set up the linear system that's going to take the measurement. So here, like X is again linearized so that it's a vector, and every so this is a multiplication, and the multiplication can be written like this in the matrix form, and every convolution can be written in terms of a matrix multiplication. And this is like just converting the convolution into a matrix multiplication. So this is like the form of the equation for a single image. Of course, you can do like undersampling or like the detection in a lower resolution, making this system smaller. But the important thing is like if I take only one image, it's not going to have enough independent equation to solve for Ex and hence X. So I'm going to need like multiple of these patterns to do that. And if I do multiple of these patterns, like I can build up this massive linear system, which at the end of the day kind of like boils down to the form, like the same form we saw before, like y equals ax, or like here, like vy equals a vx. So if I can put in something in this form, then I can solve it using the algorithm we discussed earlier, just by doing some sort of L2 and L1 mix optimization. So basically that's what we did, and these are like some of the results we got. What you see here is actually the ground truth image, or like the point scanning image, which takes a long time to acquire. And then we simulated the normal wide field image without any pattern. So TFM means the temporal focusing microscopy. It stands for the same thing without any patterns, like uniform illumination. So what you can see is like you're going to see like a lot of scattering when you go deeper. So like probably the shallower planes are fine, but when you go deeper and deeper, like you're going to see like a lot of scattering. And then we also simulated the images from the deep TFM instrument. So these are not, like still not experiments, they are just simulations. And then we used the reconstruction algorithm to do the reconstruction, and you can see like it kind of like generates an image which looks much closer to the ground truth point scanning image. But the advantage is like you can operate this at much faster speed. So, so another interesting thing is like this allows us to do like these flexible measurements. So if I image like depth, let's say like close to the surface, then I can use a less number of patterns, and hence like I can image faster at the surface. And then as I go deep, like I can just increase the number of patterns that are needed to solve the linear system. Hence it slows down. But if I look at the entire volume, like I can image the surface or like near the surface faster and then slow down as you go deep. But if I use point scanning, it's going to be the same measurement time, which is like orders of magnitudes higher in terms of the number of measurements. Yes, so the TFM can actually measure much faster. It's just single image a plane, but then it's not. It's going to have like lower quality images as you go. So these are so that was kind of like the simulated results. But these are like real experimental results. So what you see here on the first panel A is imaging a mixture of fluorescent beads through a scattering medium. And this is the TFM, or like without the pattern image. And then once you reconstruct, you can actually get a better reconstructed image. And this is for a shallower depth. And this is for about like 300 micron deep, equivalent of 300 micron deep inside the brain. And we have shown that we can still do the imaging. And similarly, this is for a biological sample, and this is actually imaging through a scattering tissue, muscle tissue, and then this is the same results for that, like but showing it side to side. You can see like the improvement that we get through this pattern excitation and reconstruction. So all looking good, and we submitted this paper, but like then the reviewers came and asking, oh, actually, like this is all fine, but these are fixed samples. Can you actually image the brain? So then in the second round, we have actually had to do the imaging of the brain. So this is an imaging of live mouse. This is the surface of the brain. And then we showed that, so these are the blood vessels. So what we do is like we inject a dye to the tail of the mouse, and then like it gets into the blood vessels, and then like the circulation. So the blood vessels lights up as fluorescent molecules. And what you see here is these blood vessels. So this is about 100 micron, and I think at the end it's about 350 micron deep, or like 300 to 350 micron deep. So we actually showed that you can perform the real, something similar to a real experiment in live animals. Yeah, so that was like good so far, but we haven't still we haven't done any like improvement in the computational side. So in this third part of the topic, like I'm going to like discuss a little bit of like how we improve this in terms of like how we take the measurements using more advanced like machine learning based computational techniques. So the basic core idea is so the entire thing right now is linear, right? The only thing that is non-linear is like how we actually do the optimization, because it has this the regularization terms and all. But the forward system is entirely like linear, and there's like no non-linear approximations when we set up the forward equations. And this is the same thing I showed you earlier. But one can actually think about this in terms of like having some non-linearity. So what you see here is instead of like thinking about some sparsity in the linear space, I can also think like, okay, so there's some like lower dimensional alpha vector that contains all the information about the image, and then I have some like function or a manifold that kind of like maps it into a higher dimension space X. So if that's the case, then alpha has like lower dimension, which means like it has lower number of unknowns. And I can set up the same set of equations, but now it's going to be a set of nonlinear equation because this psi is a nonlinear function. Then I can actually like, if the alpha dimension, I only need the number of equations as to be equal to the dimension of alpha. And if it is solvable, like then there's a high chance of being able to solve it. So like if I have this now, then I need some inverse function here. This is not really like an inverse matrix, it's an inverse function, a psi inverse, and it's a function of y. So if I can find this, like I can just like combine these equations, basically like x hat is going to be equal to psi alpha hat. And then you expand that with the a inverse, a psi inverse, a function of y. But if I think about this whole thing, it's some nonlinear function that I represent by f inverse. So now f inverse is a function of y, which gives x hat. That is what we do. So basically, if we can find some inverse f inverse function, which is a nonlinear function, that gives you from y to x, we are all set. So that's exactly what we want to do. So basically, like if you look at this framework a little closer, what I'm saying is like I have this model of A, which I model based on the physics and the microscope design itself. And then if I have like some set of data X, let's say, now I have like many X's, many images acquired previously as a database. Then I can use this forward function A to create a synthetic data set Y. So if I create a synthetic data set Y, that means like I have this like matching paired data that can be used to train some sort of a neural network F inverse. So you can do that. Like if you think about this, this is like an at the end of the day, the entire thing is an autoencoder that X goes in and X comes out. And then I can, since I know the X went in, like I can just develop some cost function to compare the reconstruction, and I can train a neural network using this data set. So that's what we did next. But we did it first for the TFM, or like without pattern case. So basically now to just recap, A has the information about the excitation point spread function, the scattering point spread function, and then the patterns, and then they are like combined in some sense in the forward model. Since we are talking about just without any patterns, this is going to be like all one excitation image, only one. And then we use like so we use like a 3D convolutional neural network as the F inverse. So the idea was like just not reconstruct one image at a time, like just reconstruct the entire volume. So this is like a 3D X and 3D X hat, or like 3D Y and 3D X hat. And then like these are the results we got after training this network. So what you see here in the point scanning, like the ground truth measurement, and this is on the test data set. And then we are interested in these small protrusions called spines. And then this is actually like a real TFM image, or like the experimental image of the same cell from the same mouse. So this was done, you image the mouse using the point scanning microscope, and then you move the mouse to another microscope, like with the wide field microscope, and then you take the image and then like look at the same region. So these are taken at 2 different times. But the network was trained on the images like this. Then these images like this were used to synthetically generate images like this to train the network. And then once this experimental image goes through the neural network, we get the reconstruction, and it seems to be like really close to the experimental point scanning image. So to show that this works, like you can't actually give some like mean squared error or like some cost, because these are not pixel matching images. Experimentally, they were acquired 2 different time points with 2 different systems. So you can't really do that. So what we did was like we just looked at the things that are interested to us. Like we manually counted these spine locations and then see like how many we can actually identify. So as it turns out, using the temporal focusing, or like without the reconstruction, we could only get about 50% of the original spines. And once we do the reconstruction, we can actually get around like 90% of the original spines. Again, like this paper is still in review. Like we submitted, and they said like, oh, this is like a very specific example. So like we have to do some other experiments showing like this is showing for a different fluorophore of the same kind of images, but now the forward model entirely changes, because like the wavelengths of this fluorophore, the wavelength that this fluorophore operates is different. So we did the same experiment, reproduced the same results, and it looks good. But the disadvantage on this system is like we have this, we can only do like shallower depths. So once you go above like 2 scattering lengths, that is around like few, maybe more than like 100 microns, then you start seeing like that this doesn't really work. This is because like when you go deep, like you really need the pattern excitation to like encode your frequency information, because like you're going to lose like a lot of information through the scattering. So obviously, like the next thing we tried right now is just build the same system but use pattern information. So this is using the encoder, building the encoder based on the physics model, which has these things, and then we have the patterns, like not a uniform image, but like some pattern excitation in the forward model. And then you do the same thing, like to try to generate synthetic data and then train an inverse neural network to do the reconstruction. So what you see here is around 200 micron deep image, but synthetically generated on the test set. So this is the point scan original image, and then this is like the temporal focusing looping image. And once we take the simulate the images of pattern excitation and then reconstructed, we can actually go into something very similar to point scan. But still, this is on test data that has not been seen by the neural network during training, but they are still on the simulations. Here, what we have done is like we have again trained the neural network for feature, like for test images of like beads. And here, this is actually an experimental image, and then this is a reconstruction done on actual experimental images. And it seems to be like promising and working well, even for the real images. So far, so good. But so far what we have done is like we used this pattern excitation, but we didn't actually like optimize these patterns. Like we just use some random or like pseudo-random kind of patterns to do the elimination. So then we wanted to actually see if there was, okay, so can we find like a set of patterns that work best for a type of images? And can we optimize these patterns, and whether will that work better than just using random patterns? In other words, I was thinking about this is like, now this is an autoencoder, but can I actually treat the encoder part also trainable? So here I'm just replicating the same results, using just random patterns. So you start with patterns like this, and this is how, like these are simulations still. This is how the measurements of like Y looks like. And then this is the ground truth, and this is like the reconstruction. So right now you're just using like some random patterns. But what we want to do is like you treat this thing as an entire thing as an autoencoder, and like make these patterns also trainable. So once you do that, these patterns actually kind of like optimize. And then this is the reconstruction we get. So far, like you can see, like if you look at the state, like it looks a little better on the, so this has to be like not fixed, but optimized encoder. And especially if you look at this letter here, this is a 5, but here it looks like a 4, but when we optimize the patterns, like we get actually the right one. So it seems to be adding something to the reconstruction when we treat this entire thing end to end. So how am I doing on time? How much time do I have left?
I've scheduled it for 1.5 hours. So including the questions, maybe we are 1 hour done.
Okay, I see. In that sense, I can actually explain this a little more. This is almost the end. Yeah. Right. So so this is how we actually do it. Like I put this slide up to give you kind of like an example or the sense of how things work inside. So this is the same framework. And A is like the forward model, and it has information about the excitation PSF, scattering PSF, and then the patterns. And then like we have this downsampling so that like you can do some compression. And then we have a noise model, adds noise into the into the measurement, and then we do some upsampling, and then use something like a ResNet architecture to do the reconstruction. But this is a shallower network still. So basically, so there are two interesting things based on the physics, right? So there's the noise model, which is actually somewhat complicated, but I'm not talk about it. But what's interesting is, or what's important is that the noise is going to depend on how bright the sample is. So if it's a low light condition, we are going to have like a lot more noise than if it was high light conditions. And then the second thing is like these patterns, like how we actually optimize for these patterns. So this entire thing is like modeled as a differentiable function. But in order, so this has to be interpretable, right? Not just like the reconstruction network here. So once I have this design of the patterns, I need to be able to go back to the instrument and implement them in the physics. So to do that, it has to satisfy the constraints of the instrument. So one of the major constraints is like we have a DMD to implement these patterns, so which means it has to be binary, it can be only ones and zeros. But if you try to like just do ones and zeros and optimize it, like this is not going to be differentiable because you have this step, right? So what we do is like we start with optimizing something which has the entire range, and it goes through a sigmoid. And then the sigmoid now has like some slope, so it maps into the range of 0 and 1. But still it's a grayscale thing because like you have the entire range. So what we do is like when we start optimizing, we have this custom sigmoid which has a larger slope, and then we start optimizing it. And when the number of epochs grow, like we start increasing M so that this becomes more and more like a step function. So at the end of the optimization, what we get here is actually more 0 and 1 or switch on and off kind of patterns. And also, so if I optimize just like the patterns it's themselves, what we are going to see is like this is going to be spatially dependent, which we didn't want. So like we want something that kind of like doesn't depend on the size of the field of view. So to get that, what we do is like we actually optimize things in the Fourier domain. So we optimize the Fourier representation of the tau, and that's where the actually trainable weights are. And then these weights are going to go to the 2D IFFT and then absolute value, and then like go through this custom sigmoid to generate the patterns. So yeah, so this is how we do it. And also like using this setup, we can test like many things. So one thing is like how sensitive this is to be like the initialization of the patterns, and also like how much downsampling one can do, so how much compression one can do, and also like how the things will have look when we do the experiment in low light conditions versus when we do the experiment in high light conditions. So all this information is actually like very important when you build a microscope. For an example, let's say I can do downsampling to like maybe like let's say 8 by 8 downsampling, then that means like that is a different sensor than the sensor I should buy if I cannot do downsampling. So all this decision can be driven through like this kind of design model, and then the microscope is going to be optimized for the particular experiment that we have in mind. So so this work is like still ongoing, but this is some of the summarization of the results. So what we have done is like we have trained like tens of thousands of networks using different hyperparameters. So hyperparameters are the ones that I showed you earlier, like how much downsampling, how much noise, like the light conditions, and the initialization, things like that. And this is just summarizing it in terms of the error, reconstruction error for the test set versus how much compression that we can get. So basically, what this gray like shaded symbols are after learning or like optimizing the patterns, and then the open red ones are without optimizing the patterns. So one trend is like, and this is for like low light condition, and this is for high light condition. So one thing that is clear is that doing optimization actually always helps, right? So it moves the symbols down. And also the size of the symbol actually represent how much downsampling that you can do. So larger size actually gives rise to better compression. And here you can see, like, if you do too much downsampling, then this doesn't add anything. So, but like in between, so that means like there is a limit to how much compression that you can do. And also another interesting thing that you see here is that some of the initialization actually kind of like matters. So you can see this open or like red triangle, which is an initialization called like Hadamard, using Hadamard matrices. So the advantage of using Hadamard matrices is they give rise to an orthogonal system. So this is like a very famous way of like designing the patterns. But what we can show is like we can do like better than that through like learning approaches. And another interesting thing is like even for low light condition, the error doesn't seems to be like the graphs looks somewhat similar, maybe a little different, but like looks somewhat similar, which means like the networks can actually do a lot of denoising. So we can operate at low photon levels or like low light conditions. Now, just to get a gist of like a sense of how we can interpret this, so I cherry picked this result. So what you see here is the low light condition, and what you see here is the high light conditions. So if you look at the patterns that the network like optimized are like totally different. So it's interesting to see like, so this highlight condition actually has a lot of like off pixels on the detector, on the VMD, whereas the low light condition has like more on pixels. Now you can see like the network actually tries to switch more pixels on so that you can get more light pass through the setup. So these kind of like results are like, I think, very interesting when we build the systems, and like we can actually see what's happening, and then we can interpret like what kind of optimizations that we can do. Yeah, so basically that's kind of like where the things are. And in summary, like what we do is like we try to build these microscopes by like modeling physics as a forward model, and then using some sort of a neural network to do the reconstruction. In future, one of the interesting things that we want to try is to see if we can do this, and then we optimize the parameters of the microscope. So one of the things that we want to do in the future is like to see if I define a specific task, like do the processing side also itself on the entire framework, and can we actually decide how we do the sampling, or like how we design the patterns or the parameters of the microscope. So these kind of like task-specific microscope systems. And also one of the major limitations in these optical systems is like they are nonlinear. Sorry, they are linear. So we are also looking into developing new like optic systems which has some nonlinearity into it. Yeah, so that's like so far it. And I should acknowledge like this is where my lab is. Like it's hosted at the Center for Advanced Imaging in Harvard. And also I maintain a little presence in MIT Laser Biomedical Research Center, and also collaborate with there. And some of this work was funded by the National Institute of Health and then Research Council of Norway. These are like actually the people who did the work. So I haven't done much work. So I just make the coffee in the lab, and then I drink the coffee. But these people actually do the work. So Shan was my previous postdoc. Now he's an assistant professor in Zhejiang University. So he did the work with 3D networks. And Nawadini was a student from Moratuwa, and she's now going to start her PhD in King's College London. And she did the work with deep TFM and then the optimization of the reconstruction networks. And Udit is a current student in Moratuwa in his final year, and he's currently working on the pattern optimization task. And these are my collaborators. Ellie is a neuroscientist working at MIT. And then Peter is an instrumentation person who's also my PhD advisor before. So yeah, that's about it. And just to keep my promise. So when we start the talk, like it was actually like the title was Turn It Up to 11. So I will actually show where the reference comes from. Let me share my screen again.
This is a talk to what we use on stage, but it's very, very special because if you
Can you actually hear the sound?
Yes, we can, Dushan. Yes.
Yeah.
you can see, the numbers all go to 11, but right across the board, 11, 11,
and most of
11,
the amps go up to 10.
Exactly.
Does that mean it's louder? Is it any louder?
Well, it's one louder, isn't it? It's not 10. You see, most blokes, be playing at 10. You're on 10 here, all the way up, all the way up,
Yeah.
all the way up. You're on 10 on your guitar. Where can you go from there? Where?
I don't know.
Nowhere. Exactly. What we do is if we need that extra push over the cliff, you know what we do?
Put it up to 11.
11, exactly. One louder.
Why don't you just make 10 louder and make 10 be the top number and make that a little louder?
These go to 11.
Yeah. So yeah. So basically, like you can ask any questions you might have now. Yeah. But the question like why we do this is because like this goes to actually 11. So that's what it is.
Hi, Dushan. Actually, I was going to ask that question. So why don't we focus on 10 and make it the top? Because as in how this compares with other optimization methods, we are essentially trying to figure out a nonlinear function here, right? So
Yeah. So basically, like here, right? Yeah. So yeah, I had the answer actually for the why we do the optimization on the patterns is just like the same thing. It just goes to 11, right? It doesn't add too much. But what we have seen is like optimizing using neural networks and like getting some of the priors that comes from the images themselves actually adds a lot of value. For an example here, to do this reconstruction, usually we need around like few hundred patterns, like some 512 patterns. But here we could do it with around 32 patterns. So that's about like order of magnitudes faster. So that's why we actually do it when you incorporate learning methods. We have seen that we can actually get into around one order of magnitude faster than the other things.
so what is the pixel number we are talking here?
Yeah, that's an interesting question. So usually it's if you look at a full field setup, it's in the order of like 1,000 by 1,000, so like 1,000,000 pixels. But these systems can have like what matters is actually like the space time bandwidth, right? How many pixels like you can read. So if you have like a large number of pixels, that sensor is going to be slow. So there is a trade-off there. But typically these images have like around 1,000 pixels by 1,000 pixels. But these regions, I think like what I'm showing here is not that big, maybe a few hundred pixels by a few hundred pixels. Does that answer your question?
Yes, thanks. So does anybody else have any questions? Feel free to unmute and ask questions, everyone.
Stop sharing.
Can you also give us an idea about the training time of the network
Mhm.
or the experimental setup?
So one of the reasons, so we have few setups, right? So for the 3D one, and then the other one, which we don't optimize the patterns, it's about 6 hours to like 12 hours. And also it depends on like how much resources we can claim on the cluster, stuff like that. But like it's in the order of less than a day.
And I had one other question. You explained about some work you did with mice in the lab there. So how does it compare with the signal processing methods that we can use, if that makes sense? I mean, these image processing based techniques in this neuroscience work, how does it compare with the signal processing approaches we can take, like multi-electrode arrays, things like that?
So we are actually just kind of like using mice because that's the kind of experiments we want to do. But my lab, and I'm also not an expert in studying neuroscience. So I think like your question is more in the order of like how the neural circuits kind of like relates to the things and algorithms we do. Is that what you asked, or?
No, my question was related to the application side actually. So because I have seen that for things like epilepsy, they use mice when they try to do drug discovery, and they're using techniques like multi-electrode array where they measure electronic signals. And yeah,
so I see. So basically, like means this where you measure the electric signal
yeah.
on cell itself. I see. That's an interesting question. So one of the advantages of doing this optically is like we don't need to do any it's once you design the mouse, that is kind of non-invasive, right? We don't have to poke through and then implement something like an electrode to measure. So there are like few techniques called like voltage imaging and then like calcium imaging that can look at how the signals pass through the neurons.
Yeah.
So these probes are like optical probes, and they are genetically encoded into the mouse. So it's more non-invasive, and also it has like higher throughput, right? So one of the things we are trying to do is like increase this size of the field of view, or the area that you can see at once, so that we can see the entire picture, looking at like hundreds of neurons, measuring these voltage signals from hundreds of neurons. That kind of like scaling up can be done when we do imaging. But if you do it with the electrodes, like you have to patch the cell, like every cell, right? Which is going to be like very difficult to kind of like scale up the type of experiments. But when we do it optically, like using imaging, you can actually scale it up if you can make these faster microscopes. But the disadvantage is like if you patch it up, like you can actually go and target one cell, and you can probably go much deeper than what you can actually do with optics. So you can look at the deeper layers of the brain.
And are you looking into applying these techniques into other domains? Maybe not images, but things like DNA sequences, maybe? Or
yeah, so like not really the imaging methods, but we have one project like we just kind of like started off looking at the DNA. So DNA sequencing is very good to kind of like profile cells, right?
not the image-based techniques. I mean, I meant the data.
Yeah.
Pattern recognition methods that you showed.
Yeah, so basically, yes. So the sequencing is very good to kind of like profile cells. And for an example, you can look at the transcriptome of the cell. That is how these proteins are expressed. That gives you kind of a way to identify the cell state. So we have one project that we are trying to like, so these are like mostly people use like things like transformers, like sequence-based machine learning methods to analyze these things. So we have one project where we're trying to do that, gives you a very good like solid ground truth to the state of the cell. So we have this one project where we try to do like do the sequencing and then do the imaging of the cells on the same cell population. And then we are trying to kind of like use machine learning to link up these features and then see like if we can actually get the same state of the cell by doing imaging without having to do sequencing. So that they kind of like, they're trying to incorporate multimodality data and then see whether we can get the same information out of it. This kind of experiments actually helps you just without, if you sequence the cell, that means like you're going to destroy the cell, like you can't use it anymore. And so you can't also study it anymore. It's a destructive method. But here, if you can do imaging, it's an undestructive method. You can just image through the state of the cell and then like see how the cell shape and the morphology kind of like relates to the internal functions of the cell. Yeah, so we are looking into some projects like that as well.
Right. So one last question I had. So how about applying these techniques in a Sri Lankan context? I mean, do you think we have the technologies we need in terms of our biology departments? Or
yeah, so I'm not really kind of like familiar with, I don't have any Sri Lankan collaborators in like who's doing the biology. But I haven't also heard of like any labs who does mouse experiments, for an example, but I might be wrong. But one thing is like these experiments are going like usually very expensive.
Right.
So for an example, if you want to build this microscope, the laser itself will cost like few $100,000. So it's about $1 million setup. So that kind of like investment, you need to have that investment if you want to do this this kind of science in Sri Lanka, which I'm not really familiar with the funding landscape of Sri Lanka, so to know like whether you have access to that kind of funding or not. But one thing that I am doing with, like I showed in the teammates slide, so I'm trying to like work with students from Sri Lanka so that we can use their expertise, and the things that we can do is like in the computational side, right? So because we don't need much resources to do that. So we can generate the data in Harvard or MIT, build the microscopes and everything. But to do all the design parts and then the data processing part, we can actually collaborate with Sri Lankan scientists and Sri Lankan students to kind of like get that work rolling. So that's, I think, where we can actually collaborate without needing to have a lot of funding.
so if I am working in Sri Lanka on this kind of data, so how do I access the data? Is it smooth?
It's usually like okay. So one thing, like, so there are most of the funding like requires us to publish the data sets, or like most of the resources when we publish the papers. So that way, like the data is actually in the like it's open access, or like if someone contacts me requesting, or can you like give us these data sets, like we have to do it. So
because they will be in order of high gigabytes, right? Maybe.
Oh, yeah, there might be there might be kind of like logistical concerns, like transferring the data as such. Yeah, but so my collaborators usually, like what we do here, the setup we have here is like we have a Harvard cluster, and we have all the data and then the computational power on the cluster, and then that kind of like I just have to give them access to the cluster so that they can do the coding on their like local computer, and then like you can actually train the networks and you do it on the cluster here. So that way, like we don't really have to transfer the data. But even if you transfer the data, it's probably in the order of like a few terabytes, which is not very difficult. You can even just like copy it into a hard disk and send it.
Yeah,
we have done it.
Yeah, I have heard that sometimes it's the case that you have to copy into hard disk and somehow send it.
Yeah, we have done it, but we haven't recently done it because like even the training is usually done on some sort of like cloud. So we don't really have to do it. We just have to have the data there. So we haven't done it before. But I think like 5 years ago, that was the way to go. Like when I was in Singapore, like what we do it, like you just copy the data and then carry the hard disk or like send the hard disk.
Yeah. Thank you for answering those questions. So let me again ask the audience. So if you have any questions, please ask. No question?
Now the one simple question. So to generate these synthesized images, we had to go through several processes, right? Like sampling, compression ratios, different kind of operations, right? So and optimization. So do you have like any validation methods to confirm your results that you generated from the system?
I see. Yeah. So interesting question. So basically, so the question is like, so we have a model to generate the synthetic data.
Yeah.
So the question is like how that synthetic data is actually how similar it is to the experimental data that we'll acquire from the setup, right?
Yeah.
Yeah. So that's actually an important question. So so what we do is so first of all, like this is what we are modeling is physics, right? So so more often, like we can actually get the model right, because physics is like physics, like it doesn't change. So if you have the right model, like and then if you have the right model and the right parameters, you can really generate very realistic looking synthetic data. But the problem often is like getting the parameters right. So that we actually do through comparing. So we have a like validation experimental data set acquired, and then we compare the statistical properties like the noise and the light levels and stuff on the experimentally acquired data set for validation of the synthetic results. And that way, like we find out the parameters. That's one thing. And then the next thing is like actually when we do the experiment, like since we do kind of like everything in-house and people are like, we have experts in the photonics side. So they are really careful in generating this data. So we actually do like proper documentation, like how much, what are the parameters that were used to generate the data sets. And those two things actually kind of like gives us a way to validate and generate synthetic data, which are really very realistic compared to the experimental data. And then the final thing is like more often like it's very easy to see, like validate or like test, because if you train a network with wrong data set, it will not work for the experimental data. So that we have to do at the end. We have to demonstrate that the inverse model we train or the neural network we train on synthetic data works well with the experimentally acquired data. So in that sense, like what we do is like we actually measure the same sample using the ground truth imaging modality as well as the test imaging modality, and then we test the reconstruction on that test data compared to the ground truth imaging modality.
Okay.
So there are like
Yeah.
few layers of validation. Yeah, so if you don't have any questions, like maybe I'll ask some questions for the faculty here, if we still have some time. So how is it kind of like the research landscape in Sri Lanka, and like what do you guys normally do? Do you collaborate with people, or how is it like? Damayanti, I think you're on.
We have staff members specializing in different research areas, Dushan. So to talk about myself, so my area of interest has been computational biology, because my PhD related with pattern recognition and DNA sequencing data. So I have looked into the RNA-seq or the gene expression analysis and also things like genomics data analysis, things like that. And I happen to work with the neuroscience laboratory at Melbourne. So at the moment, I mainly collaborate with Taiwan and also have a couple of projects going on. And we are looking at, I'm looking at image processing techniques also. But Upul is majoring in image processing work in our department, I would say. But we have a very good team in computer vision side. And if you check our department website, the information are there, all the information are there. And yeah, and also I have seen, of course, as the trend goes, there are a lot of interest from the students to go into this pattern recognition or into the image processing side. And so therefore, when we are teaching, for example, I am teaching the machine learning course this time. And the artificial neural networks course. So when you are doing teaching also, we try to, one thing we try to do is for them to see the advantage of these machine learning techniques in terms of applying them in different domains. And the other thing is the fundamentals behind these techniques. So that is about teaching the courses. And there are students who are looking, they're trying to pursue maybe jobs and internships in this area. There are a lot of students with that interest as well. So personally, I am quite interested into this area, I would say, because it's something I have worked in. Yeah, so that's about myself. So it would be very nice if we could if we could do some collaborative work. Yeah.
Yeah, you should definitely talk. So what I'm looking into is actually for the project I said, like I'm looking into do some RNA-seq as well. So it would be really good to talk to you and then see if there's something that mutually interesting to do.
Yeah, yeah. Upul, would you like to add something?
Yeah, I think you covered most of the things, and he also know about me, so he has some idea. Usually we have experts in like I think Sampath Digal. We have many faculties who are working with image processing and stuff, and yeah, and we have some final year projects. They are like using these techniques, and they have produced some interesting results, as you know now. Yeah, so we have that kind of caliber here, and we are doing mostly with data because we don't have like facilities like there. So we are usually working with a lot of data, and yeah, that's the sort of work we do here.
Yeah, nice. Yeah, Upul, I know, like we also have sort of like a running collaboration right now. So, yeah.
Okay. So if there are no questions, I think we can wind up for the day. So before that, let me again thank Dr. Dushan Wadduwage for joining us and zooming in into microscopy level. And I believe it has been very useful for everyone to start off in this field, because I think a lot of them who are here, they are trying to start off in this field. So it gave a good overview of about what sort of things we can do. And also, yeah, it gave an idea about the whole process, I would say, starting from the experimental level and going into the data analysis level. So I think that was I believe it was quite useful for the students to start off in this kind of work. So hopefully they will be able to use that knowledge in their coming projects, maybe in their final year projects, and also in their careers. So thank you so much, and we'll keep in touch. Thanks, Dr. Dushan.
Yeah, thank you so much. And thank you very much for the invite, and also like thank you everyone for attending and like bearing through.
Okay then. Okay, I'll see you then. Bye everyone.
Bye. Good night.
Good night.
Thanks, Navada. Bye.